All right, so before I really start talking about this particular slide, I'm going to go back to the notes that we've already covered this semester and make it clear as to what you need to still be responsible for this particular section of content. So historically in a textbook, you would see structure of DNA that we have here, okay, all the various slides that covered the structure basically, and you would see the content that we're about to cover, starting with that slide that was in pink all together, right? So just kind of to give you a little bit of FYI why I'm doing this differently. So you need to understand the structure of DNA in order to understand the processes that we are about to talk about. So you still have to know all of that, adenine with thymine, guanine with cytosine, and the DNA molecule. Historically, textbooks cover structure, they then talk about DNA replication, and then they talk about protein synthesis, which is where we're going. In the grand scheme of things, a lot of people tend to think replication has to happen before proteins can be made because of the structure of textbooks. And so instead, we covered structure of DNA, we covered replication, we covered cell division, right? We're stepping away from cell division briefly, and we're going to go on to protein synthesis. We're going to come back to cell division. Okay, so we're going to come back to cell division, basically the cell cycle, okay, that this slide you're seeing here leads into the mitosis notes. We're going to come back to that, and we're going to cover the other type of cell division, meiosis, after we cover where we're going next. Okay, so all of that being said, this slide, okay, is the 6th slide in your first notes packet for this semester, okay? That particular notes packet starts with this slide, okay? So, So for this particular unit, you still need to note this slide all the way through this slide. And those first 6, we're going to keep on using, okay? We're going to take a break from the cell division stuff, like I said. And so we're going to transition to needing to know structure of DNA and then heading into where we're going next. Here we go. All right, so notice this says DNA, RNA, and protein synthesis. Tells you to look back at those notes concerning structure of DNA, basic vocabulary, history, and replication. Well, we're not really going to need the replication and the history so much with where we're going next, but it does help to kind of mesh all of this together. So notice here, and these are in white. As far as I know, that contrast isn't great, but that's because I was running out of black ink apparently. But it says that DNA is used to make mRNA. We're going to talk about what that means in just a minute. mRNA is used to make proteins. Proteins control the expression of genetic traits, along with serving many other roles in the body. So again, going back to last semester, proteins control everything. Okay, they're super critical to life functioning. I'm not going to try to explain any of those diagrams because they're just too hard to see. Okay, so we know DNA is in the form of the double helix. Adenine always bonds with thymine in the center of the double helix, and guanine always bonds with cytosine in the center of the double helix. And sugars, deoxyribose sugar and phosphate groups alternate along the backbones. RNA stands for ribonucleic acid. Okay, so the sugar is structured differently, okay, and we don't get into that too much, but the structure of DNA, your ribose sugar, RNA is single-stranded, and there is never thymine in RNA. The base that replaces thymine is known as uracil. Okay, so uracil is literally going to be in the place of thymine. Okay, there are 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. I'm going to actually start with ribosomal RNA, shortened to rRNA. We do very little with this in this course. Okay, basically what you need to know, it's a component of the ribosome and it helps with protein synthesis. That's about all we do with it. Okay, messenger RNA is shortened to mRNA. That's what DNA is going to produce, and we're going to get into that process in just a few minutes. It's going to carry messenger RNA is going to carry the instructions for making proteins from the DNA molecule in the nucleus to the ribosome. It's kind of seen as a little throwaway copy of the genetic information stored in the DNA, and those different sequences of bases along the strand of DNA that would make, or strand double-stranded, that would make up a chromosome, or strand of chromatin. Transfer RNA is shortened to tRNA. The job of the transfer RNA molecule is to bring the amino acids to the ribosome. It's like a little interpreter molecule. All right, so to make a protein in the cells of our bodies, DNA is going to code for that protein, right? But DNA is stuck in the nucleus, as we can see here. So this is the nuclear envelope. There's the DNA double helix, okay? And so DNA can never leave the nucleus, right? So instead, DNA is going to have to unwind and unzip just like it did for replication, right? But it's going to have to unwind and unzip this time in order for that information that's stored in the DNA molecule to be transcribed into a new form, right? So this is copying the information from the DNA into a new form, the messenger RNA, okay? A good analogy for this is if I am talking and I tell you to write something down on your paper, that you take my spoken word and you transcribe that into written words on your paper. That's what transcription is, okay? So just for another little analogy. So transcription has to happen in the nucleus before a protein can be made, right? And so then again, that messenger RNA, it's going to be a copy of the information stored in the DNA. It's going to go out of the nucleus via these little nuclear pores, those little holes that you see all over the nuclear envelope, okay? It's going to go to the ribosome, and at the ribosome, that messenger RNA is going to be read, okay, to make a protein. Okay, and that's basically everything that's up there. So for your simplified process, DNA is going to be unwound and it's going to be unzipped via enzymes. The mRNA copy is going to be made from the DNA according to base pairing rules. So now, if in DNA we had, let's just put all 4 bases, okay, in a sequence, let's just give the other half of the double helix. This would be the double helix there. Okay, oops, you can't even see what I'm writing, haha. Okay, so if that's your double helix and that these bonds are broken and that unwinds and unzips, if this is the DNA sequence, hang on, then in messenger RNA, the mRNA sequence would be this, okay? So then that sequence of mRNA is what's going to leave the nucleus, okay, and go out of these nuclear pores. Right, it's going to go to the ribosome specifically. That particular organelle is going to synthesize proteins, and then the DNA strands, let me go back to here, that we can see here, they're going to rejoin. Okay, again, those weak hydrogen bonds are going to form again in the middle, and it's going to coil up again, following the transcription process. I'm going to stop there with this particular process. If you have a question, let me know.